Hello friends, in the previous video we were talking about the normal physiology of gastric acid secretion in the stomach, but in today's video we will be more talking about the exact mechanism of synthesis and release of gastric acid from the parietal cell. We will also see the regulation of gastric acid secretion by the parietal cell. We will see how gastrin, how prostaglandin, how histamine, somatostatin and acetylcholin regulate the gastric acid secretion from the parietal cell. Okay, so before understanding about the mechanism, first of all let me tell you this is basically a parietal cell, and this is the lumen of GIT. If this is the lumen, obviously this will be the luminal side, and this side of the cell will be basolateral side of the cell. Luminal side I have made very straight, but in fact it is not that much straight, it is folded just to increase the surface area, and this folded side is basically called as canaliculus. Okay, canaliculus basically increases the surface area of parietal cell. Well, there are certain proteins that play very important role and very important role in the secretion of gastric acid from the parietal cell. Some are present on the basolateral side and some are present on the luminal side. So if we talk about the proteins that are present on the basolateral side, they include potassium channel, they include sodium potassium ATPase pump, they include chloride channel. Similarly on the basolateral side there should be sodium potassium ATPase pump and bicarbonate chloride exchanger or antiporter, because both of the substances are moving opposite to each other, so it will be called as antiporter. Okay, now what will happen, let's start from this sodium potassium ATPase pump. This pump is basically responsible for taking 3 sodium out of the cell and 2 potassium into the cell, and for this purpose it will use 1 ATP. By using 1 ATP it will transport 3 sodium out and 2 potassium into the cell. So it means the intracellular concentration of potassium channel will increase. Similarly on the luminal side you can see the potassium channel, and this potassium channel is simply responsible for the passive diffusion, just for the transportation of potassium ion from higher concentration to the low concentration. In this case you can see that concentration of potassium ion will increase. Why? Because potassium is continuously moving into the cell. When the concentration of potassium ion will increase, this potassium will start moving out of the cell into the lumen of GIT by using this potassium channel. Okay, now leave this point here, we will come toward this point later. First of all let me tell you that the carbon dioxide that is coming from the blood and entering into the cell, it will react with the water and it will form carbonic acid. Carbonic acid being acid, obviously if we look at the definition of acid are those substances which will dissociate and produce hydrogen ion in aqueous medium. So obviously inside the cell there is aqueous medium, so this carbonic acid will dissociate into hydrogen ion and bicarbonate ion. Now this hydrogen ion will use this hydrogen potassium ATPase pump. With the help of this hydrogen potassium ATPase pump, this pump is basically responsible for taking hydrogen out of the cell and taking potassium into the cell, and again it will use 1 ATP for this purpose. It will use 1 ATP. It means by using 1 ATP it will transport 1 hydrogen ion out of the cell into the lumen and will transport 1 potassium ion from lumen into the parietal cell. Clear? Okay. Now in this way the concentration of hydrogen ion will increase in the lumen. Now what happened with the bicarbonate ion? Bicarbonate ion will use this bicarbonate chloride ion exchanger, and in this way this exchanger is basically responsible for taking bicarbonate ion out of the cell and taking chloride ion into the cell. In this way obviously the concentration of chloride ion channel will concentration of chloride ion will increase. If the concentration of chloride ion increases, this chloride ion will simply use this chloride channel that is present on the luminal side of the parietal cell. By using this chloride channel, the chloride ion will diffuse out of the cell into the lumen, and here you can see chloride ion and hydrogen ion, they will react to produce hydrochloric acid HCl. Okay, so this was the synthesis and release of acid from the parietal side. Now let's see how this secretion is being regulated by different substances. So here you can see on the basolateral side there are receptors CCKB. These are the receptors for gastrin, and in the previous video I have talked about that gastrin, histamine and acetylcholin, they will basically increase gastric acid secretion. Okay, if you want to know more about these cells and their secretion, you can visit my previous lecture. Similarly, we have seen that prostaglandin and somatostatin basically inhibit the gastric acid secretion. Now let's see how it happened. Basically, now let's start with the histamine. When histamine released from the enterochromaffin cells, they are present just near the parietal cells in the GIT in the gastric pits. So when histamine will act on the H2 receptor, what will happen? H2 receptors are linked with the GS protein, and when GS protein get activated, what will happen? It will ultimately activate adenyl cyclase, an enzyme adenyl cyclase. This adenyl cyclase will convert ATP into cyclic AMP, adenosine triphosphate, a nucleotide. It will convert this ATP, this nucleotide, into cyclic AMP, and the concentration of intracellular cyclic AMP will increase. When the concentration of intracellular cyclic AMP will increase, it will ultimately activate another very important enzyme that is called as protein kinase A. Now if you look at this word kinase, what is the role of kinase enzyme? Kinase enzymes are basically responsible for the phosphorylation. They are responsible to attach phosphate group on other proteins. So similarly, these protein kinases A will activate these Gastrin protein. Gastrin protein, it's a member of Gastrin protein is a member of cytoskeleton protein. What is the role of Gastrin protein? I will explain after 1 minute. So what I was talking about, intracellular cyclic AMP concentration will increase, and ultimately protein kinase A will be activated. And this protein kinase is responsible for attachment of phosphate group on other proteins, and those proteins will get activated. Okay, after attachment of phosphate group, other proteins will get activated. So this protein kinase A will activate Gastrin just by attaching a phosphate group. So after the attachment of this phosphate group, this Gastrin protein is, I am taking it as an example. Gastrin protein is basically member of cytoskeleton protein. So there are some other proteins that will be activated by this protein kinase A. Okay, and when these cytoskeleton proteins, which include Gastrin protein, will get activated, you know what is going to happen? This tubulovesicle, let me write its spelling. Tubulovesicles. These are the vesicles present in the parietal cells. Now these vesicles hold hydrogen potassium ATPase pump, and these vesicles hold potassium channels. Okay, so it means obviously after the for example, let me explain. So here is the nucleus of this parietal cell, and if this is a nucleus, there must be some DNA. Yeah, okay, if there is a DNA, this DNA must be having genes for this hydrogen potassium ATPase pump, because they are protein in nature. Every protein will be having its specific gene, okay, and potassium channel. So assume that these are the genes. This gene is basically for the hydrogen potassium ATPase pump, and this gene is basically for the potassium channel. So what will happen after the transcription? Messenger RNA will be formed. After the transcription of these genes, messenger RNA will be formed. Then this messenger RNA will come out of the nucleus, and it will attach on the ribosome. These messenger RNA will attach on the ribosome. Now what will happen? Proteins will be formed, and what will be these protein? From this gene, hydrogen, potassium, ATPase pump will be formed, and from this gene, potassium channel will be formed. Now this proteins, so hydrogen, potassium, ATPase pump and potassium channel will be placed over the tubulovesicles, organelles. These are the basically organelles present in the parietal cells. So they will be placed on the tubulovesicle. Now what will happen? These tubulovesicles then will be transported, fused with this apical side of the, they will be transported and fused with the apical side of the parietal cell, and when they will get fused over the plasma membrane, they will get activated and they will start transporting hydrogen ion out of the cell and potassium into the cell. Getting it? Okay. Now what will happen? These Israel protein that has been activated by protein kinase A is basically responsible for increasing the transport of, increasing the fusion of these tubovesicles with the plasma membrane of, with the plasma membrane or with the canalicula of parietal cell, with the plasma membrane of the parietal cell. Again I will repeat, histamine will bind over there, adenyl cyclase will get activated, adenyl cyclase will convert ATP into cyclic AMP, cyclic AMP will activate the protein kinase A. Protein kinase A will activate cytoskeleton proteins, for example Isrin, by placing phosphate group over them. When these proteins will get activated, these proteins are basically responsible for the transportation and fusion of tubulovesicles with the plasma membrane of the parietal cells. Now these tubovesicles will be having hydrogen, potassium, ATPase pump and potassium channels, okay. Now similarly, gastrin and acetylcholin, they will also activate these Isrin protein, but they will not follow this pathway, they will follow different pathway, that is IP3 and calcium ion dependent pathway. IP3 and calcium ion dependent pathway. Obviously these receptors are basically linked with the GQ protein, and GQ basically activates the phospholipase C, and phospholipase C will convert inositol diphosphate into diacylglycerol and IP3. IP3 will act on the smooth endoplasmic reticulum and increase the calcium ion efflux into the cytoplasm. When the calcium level will increase in the cytoplasm, it will bind with the clomidulin, and calcium clomidulin complex will be formed. Now this calcium clomidulin complex can activate different kinases, and similarly diacylglycerol can activate protein kinase C. So by following this pathway, they also will activate this Isrin protein or other cytoskeleton protein. Okay. So what will happen after the activation of this cytoskeleton protein? The transportation and fusion of this tubovesicle will increase. Now we know that prostaglandin and somatostatin, they basically decrease the acid secretion. They basically decreases the acid secretion. Now let's see what and how these how this works. Basically these receptors, prostaglandin E2 and somatostatin receptor, they are basically linked with the G inhibitory protein, GI protein, and GI protein basically they will inhibit the adenyl cyclase. They will not activate the adenyl cyclase like histamine was doing. They will inhibit the adenyl cyclase. If adenyl cyclase will be inhibited, the conversion of ATP to cyclic AMP will be decreased. If this concentration of cyclic AMP decreased, activation of PKA, protein kinase A will be decreased. If activation of protein kinase decreases, activation of Isrin or other cytoskeleton protein will decrease. If the cytoskeleton protein and Isrin activation decreases, what will happen? Transportation and fusion of tubovesicles will decrease. If the transportation of tubovesicle decreases, what will happen? The movement of hydrogen ion and potassium will decrease. So less hydrogen ion will go into the lumen, so it means acid secretion will decrease. So this is the reason prostaglandin and somatostatin basically decreases the acid secretion. Okay. So it was all about the cellular mechanism related to the synthesis, release and regulation of acid secretion from the parietal cell. I hope you like my video, please don't forget to like, share and comment on the video, and don't forget to subscribe my channel so that you may get notification for the future video. In next video we will be talking about the pathophysiology of ulcer. Okay, we'll talk about how H. pylori causes ulcer, and all the molecular aspects will be discussed. Similarly, we'll also talk about how NSAIDs and other pathological reasons causes the ulcer. Okay, thank you so much.